Immune evasion, an illustrative synopsis. Pause illustrations and text for detailed study. A pathogen is an infectious microorganismal bacterium, protozoal parasite, or virus causative of disease in a host organism. Infection results in the host innate and adaptive immune system responses detailed in prior presentations, as well as the various virulence characteristics and mechanisms manifest by the particular pathogen. The restoration of immunological and physiological homeostasis in the host is dependent upon the type and degree of infection, the efficacy of the prophylactic and therapeutic therapy received, as well as the sufficiency of host immune responses. Various pathogens possess the inherent capability to effectuate immune evasion mechanisms, resulting in host cell invasion and colonization, exploiting these cells to access the tissues targeted by the pathogen. This process of immune escape, also referred to as antigenic escape, immune escape, or escape mutation, occurs when the immune system of a host is rendered unable to recognize and eliminate an infectious pathogen. The origins of this process are environmental and genetic. The immunomodulatory mechanisms of bacteria and viruses must overcome identical host cell immune mechanisms and have developed parallel mechanisms to neutralize same. Particular bacterial and viral diseases may possess a linkage whereby host immune defense vulnerability is caused by a diverse pathogen, as is the case when infectious influenza predisposes a host to pneumococcal pneumonia infection, morbidity, and mortality. Immune escape also plays a role in resistance against vaccination. Since a vaccine does not encompass every strain of the particular pathogen, the antigenic DNA recombination resulting in the diversity of a pathogen may enable it to resist the intended effects of even the most currently produced, administered, and specific vaccine. The antigen may have acquired the capability to effectively target disease-causing pathways diverse from those specifically targeted by the vaccine, rendering it ineffectual. Viruses and bacteria demonstrate a variety of anti-immune response mechanisms. Antigenic hyperavailability involves error-prone replicase expression and evasion from antibody and T-cell recognition ability in viruses. This characteristic is observed in bacterial variations between strains, various surface structures, pili, lipopolysaccharides, LPS, and outer membrane proteins. Blockage of acquired immunity is via major histocompatibility complex MHC1 and 2 downregulation, antigen presentation and proteasome blocking, and the prevention of immune response genes in viruses. In bacteria, this action occurs via immunoglobulin A proteases, complement degradation proteases, and antigen presentation blocking. Blocking of small antimicrobial molecules can be inhibitive and preventive of antiviral RNA silencing and independent nitric oxide synthase in viruses. This capability in bacteria results for cell surface alterations for the avoidance of peptide insertion, sensing of small molecules capable of triggering defense mechanisms, and peptide transport by means of pumps. Inhibition of complement occurs via complement cascade soluble inhibitors and fragment crystallization FC receptors in viruses. The FC tail region of the antibody interacts with host cell surface FC receptors and complement proteins for antibody activation of the immune response. In bacteria, capsules and long-chain lipopolysaccharides are produced to avoid complement deposition and membrane attack complex MAC function. Protease degrades complement, also serving this function. Interference with toll-like receptors in viruses occurs by the blockage or hijacking of the TLR signal and prevention of its recognition. In bacteria, alteration of TLR ligands decreases recognition, TLR binding to lessen inflammation, and injection of effectors for downstream inflammation inhibition occurs. Intrinsic cellular pathway blockage is inhibitory of RNA editing and regulatory in ubiquitin IS-glyation pathways in viruses. Alteration of transcriptional programs and ubiquitin pathways is observed in bacteria. Inhibition of chemokines, cytokines, and interferons occurs via blockage of secondary antiviral gene induction, inhibition of ligand gene expression, interference with effector proteins, and ligand and receptor inhibitors in viruses. This function occurs via alternate pathway activation, inflammatory pathway blockage, and protease secretion for degradation in bacteria. Modulation of apoptosis and autophagy in viruses involves inhibition of acceleration of cell death, blockage of death signaling pathways, downregulation of death receptors or ligands, inactivation of death sensor pathways, and the scavenging of free radicals. This occurs in bacteria via apoptosis inhibition, activation of death signaling pathways, and the alteration of apoptosis signaling pathways. Modulators or toxins secreted by some viruses may function as virokines or viroreceptors, respectively mimicking virokines and viroreceptors. Some toxins and proteases are manifest by bacteria for this purpose. Pathogen surface modulators of viruses may be adhesion molecules, coagulation regulators, complement inhibitors, and immune receptors. In bacteria, adhesins and invasins, lipid A of lipopolysaccharides, carbohydrates like capsules, and outer membrane proteins may serve this purpose. Subversion or killing of immune cells and phagocytes involve the alteration of immune cell signaling, effector functions, or differentiation, expression of superantigens, infection, and killing of immune cells, such as dendritic cells, DCs, antigen-presenting cells, APCs, lymphocytes, and macrophages, and inhibition of cytotoxic T cell, CTL, and the natural killer, NK cell, killing in viruses. This function in bacteria involves superantigens, blockage of antigen presentation, and inflammatory pathways by the injection of effectors, immunoglobulin A proteases, as well as replication within and overrun immune cells. Viral latency and infection of immunocompromised tissues in viruses, as well as bacterial phagolysosomal fusion avoidance and phagocytosis inhibition, can result in evasion from immune surveillance. Coronavirus innate immune evasion illustration. The innate immune response to COVIDs is activated upon detection of viral pathogen-associated molecular patterns, such as double-stranded RNA, via host pattern recognition receptors, PRRs, such as retinoic acid-inducible gene 1, RIG1. Following viral recognition, transcription factors including nuclear factor kappa B, NF kappa B, activator protein 1, AP1, and interferon regulatory factor 3, IRF3, are activated and translocate to the nucleus, where they induce the expression of interferons. Both Middle East Respiratory Syndrome coronavirus MERS-CoV and severe acute respiratory syndrome coronavirus SARS-CoV through their MN non-structural proteins NSP1, 3B, 4A, 4B, 5, 6, and papain-like protease LP-Pro have developed mechanisms to interfere with these signaling pathways. This alters the cytokine secretion profile of infected cells to enhance the recruitment of myeloid immune cells over NK cells, which in turn produce more cytokines, creating a cycle of inflammation that damages the lung. Many of these processes are likely conserved in SARS-CoV-2. The perforin protein is released by immune cell natural killer cells that destroy target cells, forming pores in the target membranes for this purpose. Granzymes are serine proteases released by cytoplasmic granules in cytotoxic T cells and NK cells, inducing apoptosis in virus-infected cells. Fibrin is a white insoluble elastic protein essential to normal blood clot formation. More presentations coming soon. Thanks for watching.